Good morning, everyone. Welcome to another wonderful High Rounds. I think even more wonderful because we have a speaker that is new to us and not so new anymore, as he just reminded me, to UCSD. I think a really exciting person to come and join UCSD, and this talk should be fabulous. Dr. Michael Corley is a tenured associate professor of medicine in the Division of Geriatrics, Gerontology, and Palliative Care at UCSD, where he leads Geroscience research at the Salmon Rosenstein Institute for Research on Aging and the UCSD Center for Healthy Aging. He earned his MA and PhD in behavioral neuroscience from the University of Hawaii and completed postdoc training in epigenetics, immunology, and infectious disease at the John A. Burns School of Medicine. Prior to joining UCSD, he was an assistant professor of immunology and medicine at Weill Cornell Medicine, where he continues to collaborate on projects exploring the interplay of epigenetics and HIV. Through his transdisciplinary work, Dr. Corley aims to identify novel epigenetic markers, therapeutic targets, and gerotherapeutics that improve the health spans of people with HIV. And with that, I will turn it over to you, Michael. And thanks for joining us today.
Yeah, thank you so much, Jill, and happy Friday, everyone. And yeah, today I'm quite excited, I think, to share some of the more recent work that our lab has been pursuing, and really, I think quite excited to plug into the incredible research going on here at UCSD. Today's talk, I'm really going to talk about our work looking at HIV and aging and kind of the general idea of zooming out a little bit and thinking even further within the UCSD community, and particularly within San Diego and La Jolla, on this real exciting, I guess, area of research that has always been there, but really I think has recently kind of received even more attention in terms of the aging research landscape. And kind of the concept of how a human retrovirus, such as HIV, can advance this geroscience agenda. So for today, I wanted to spend a little bit of time about the field of geroscience and what this field is kind of doing currently. Our basic science research in terms of how we've learned about some of the mechanisms of how cells are orchestrating their gene programs, and how the geroscience field has used this as a pretty well established now and emerging biomarker of aging. And the second part, I'll also kind of build a little bit more in terms of how we've learned about HIV as an exogenous retrovirus that has kind of informed our understanding, and really tried to collaborate with groups that are interested in retroviruses, so endogenous retroviruses that have invaded the germline, and how we've used some of our insights from HIV to look at some of these aging programs that are in our genomes. And then lastly, I wanted to spend today talking about the most exciting area of our research program, which we're really harnessing HIV clinical trials to inform geroscience-guided therapeutics. And the idea here is taking essentially therapeutics that were initially designed either for HIV cure or HIV comorbidity treatment, and looking to see whether those therapeutics potentially are modifying some of these aging biomarkers or hallmarks that people are interested in trialing in the general population. And really there is just to try to get some evidence to fill in the gap where there isn't evidence currently from rigorous clinical trials for the aging field. In terms of the geroscience field, the field is really the study of genetic, molecular, and cellular mechanisms that make aging a major risk factor and driver of chronic conditions and diseases of older people. And the general idea is, rather than focusing on a specific disease and mechanism related to something as dementia, the idea would be thinking about potentially those mechanisms that are driving the aging process, and those potentially being a major driver of leading to Alzheimer's disease and other comorbidities, which usually present together in terms of older people, and particularly within people living with HIV. And the geroscience hypothesis states that slowing the aging process can improve health and delay the onset of chronic diseases. So really this concept of prevention, if you're able essentially to intervene and potentially impact some of these biological hallmarks of aging, this could slow down the aging process and essentially prevent the disease outcome from arising. In terms of processes, we know that accelerate aging. Environmental factors have been widely shown across multiple studies, epidemiological studies, those studies where people are followed in these large cohorts here within the US or even globally, that contribute to the effects of the aging process. And I think for the group here, everyone's well aware of the impact of infections and other particular factors that are drastically altering our immune system and essentially leading to these immunological changes that, in essence, are really driving an increased biological aging process over time. And recently, too, a lot of this work has also shown that some of these latent infections are starting to rise in older ages. And there's a lot of great data coming out showing that some of these infections that were latent that start to arise are now being linked to the incidence of particular neurodegenerative diseases in particular. In terms of HIV, I would say the most remarkable success, as everyone's well aware on the call, has been antiretroviral therapy. If you look at it from the aging landscape, it's been the best aging therapeutic to increase an individual's lifespan. In terms of the emergence of antiretrovirals, I think another area we're quite interested in today, I won't spend time on it, is in terms of looking at HIV prevention and the impact potentially of PrEP and those therapeutics in terms of people that are not living with HIV and how those may be impacting the aging process, I think is another area that our group is interested in working on. In terms of those people that are living with HIV and antiretroviral therapy, we do know across multiple well-funded studies sponsored by the NIH, a lot of work here at UCSD as well has contributed to this, that the risk of age-associated diseases is higher in people on antiretroviral therapy. Despite them having a very typical lifespan, their health span is drastically impacted. So the number of years they live without disease is reduced compared to the general population, suggesting that there are some potential mechanisms and lingering potential inflammatory processes or essentially impacts of the virus that is still in the body that essentially is driving the aging process in the background. In terms of framing this from this idea, as I said before, the geroscience field and the mechanisms, so essentially focusing on these hallmarks of aging, such as gut dysbiosis, genome instability, telomere attrition, epigenetic alterations, which is a large focus of our lab, loss of proteostasis, disabled microautophagy, dysregulated nutrient sensing, mitochondrial dysfunction, cellular senescence, stem cell exhaustion, chronic inflammation. These are all processes that essentially the field of HIV and a lot of great work has shown are perturbed in people living with HIV. So essentially, if you look at the landscape of geroscience and these hallmarks of aging, you can find evidence across various cohorts that essentially these processes and mechanisms are quite altered in people living with HIV, suggesting support for the idea that people living with HIV potentially have, or a subset have, accelerated aging or attenuated aging that occurs. And when we frame this in the geroscience hypothesis, the idea is that there's this very unique potential to benefit from geroscience interventions in people living with HIV. And here I'm just showing you essentially the idea that the geroscience field has, showing men and women, what's the lifespan, and then the incidence of comorbidity on the y-axis. And in green here is the geroscience hypothesis, suggesting potentially that you may be able to intervene and alter this line of the aging process and prevent the incidence of comorbidity from occurring earlier in life and therefore achieving a longer number of years living with a better health span. If you think of people living with HIV, a subset of people living with HIV potentially could even have a greater room for improvement if interventions can be identified and administered at an earlier time in life to prevent essentially this increased incidence of comorbidity. In terms of our work, a large part of our work has been built on the geroscience side of the field and applying a lot of this within the HIV field. We've been quite interested in using readouts from the immune system to measure the aging process. There's been a lot of recent activity in this space, building on an initial work that was developed here at UCSD from Trey Eideker's group, and then also at UCLA, Steve Horvath, who has received a little bit more popular press in terms of these epigenetic clocks and biological markers of aging. These have been commercialized. So these are tests essentially that you can take a blood sample and then measure these DNA methylation or covalent modifications on our genes or DNA. And these allow essentially a very accurate prediction of how old you are. Here I'm just showing you a summary table of the number of markers that they measured to be able to estimate with very high accuracy what they've essentially used as an outcome measure. The very first development of these algorithms or biological age predictors from blood samples and tissues showed essentially that they could predict how old someone was chronologically, how many years you've been alive based on your calendar birth date. And then subsequently, these other algorithms and what we've seen in the popular press in terms of these next generation biological age predictors are just using a ton of other metadata in their outcome measure essentially to predict mortality risk. So if you know essentially an individual or cohort of 1,000 people or more that have known mortality data, you can include that in the estimation models and essentially predict the time to death. So this is in reference here to this grim age marker that's been suggested. And in essence, we've been using these as ways to kind of proxy and look at cohorts of people living with HIV and potentially gain insight into interventions that may be moving the needle or having impact on the aging process. And the way that these have been described, as I had said, is just essentially chronological age, how many years you've been alive. Biological age tracks how well your body has handled those years or age at a cellular level over a lifetime. And essentially looking at the difference between your actual birth date and then your biological age has allowed kind of these links to various outcome measures people have been interested in terms of if you're above the line here or this age acceleration that occurs. This is suggestive of an accelerated age process in those individuals that are essentially below the line having a slower biological age at that time point that they've been measured or assessed. Um, there's also been some work to look at the pace of aging. So they've trained models where they were looking at aging rates over time and then have been able to tell you at that time point speedometer of aging in terms of these age estimates as well. And a lot of this work was initially developed and funded by the NIH, essentially taking all of the public metadata that was available and deposited in these genomic repositories for DNA methylation. And typically, in de identified data, one of the major variables that's included is chronological age. So you can go currently right now onto the NIH website and download these methylation data sets and get over 5,000 individuals spanning chronological age from birth all the way out to over 100 years, and then use essentially some basic models such as logistic regression or other deep learning kind of AI models to essentially predict these age outcomes. And essentially, this is kind of the creation of all these epigenetic clocks that we're seeing kind of percolate through the literature, but also in the popular press in terms of estimating how old you are biologically. This is just summarizing kind of where the field stands currently from a very nice recent review article. As you can imagine, there's been quite a lot of activity in this space to kind of build a better clock. So I've referred to these groups and labs as clock builders. A lot of this work has really been interested in kind of developing the best predictive models. We've been quite more interested in terms of looking at mechanisms that may be altered or signals. So as you look across the gamut of these epigenetic clocks, what we start to see is that each of them is measuring a different facet of the aging process. Some of them have to do with cell division genes. Some of them have to do with DNA repair and damage genes. Beautiful work here recently developed a clock showing a very nice evidence of somatic mutations occurring. So these are essentially genomic changes where methylation positions are measured or nearby them. We've been quite interested in terms of cell type composition changes. Those on the call are well aware in terms of T cell biology. But one of the main findings very early on within the aging field is the loss in naive T cells as we age. So you can imagine some of those clocks that are not accounting for cell type heterogeneity are really picking up on these immunological T cell exhaustion and loss of naive cells as individuals are aging. And you can imagine as well in the context of HIV with CD4 loss and recovery, this is a major component too in terms of some of those clocks that are proxying that particular measure. But in essence, what these clocks and these estimates have allowed us to do is essentially proxy a lot of these aging biological processes, in essence, from one kind of data modality that is measured potentially from a blood sample or a tissue sample from a person. In terms of the literature regarding HIV and the application of these clocks, I think one of the things that needs to be emphasized or appreciated is essentially the development of these clocks from Steve Horvath and the UCLA group and others at UCLA really relied on blood samples that were provided from HIV. And what they found in one of their early studies applying these clocks when they were first kind of being reported was that in a small subset of people that they had looked at that were living with HIV and those living without HIV, by these biological age estimates or epigenetic clocks, there was support based on these measures that essentially people living with HIV were older than their counterpart of a similar chronological age. And the estimate appeared, in this early study to be around 5 years greater in terms of their biological age compared to their chronological age. So some of the first evidence that these epigenetic age clocks were picking up on some of these things that the HIV field had known in terms of looking at comorbidities and other mechanisms related to the immune system and aging processes in terms of the geroscience field. In terms of our work, we got quite interested when I was a postdoc in Hawaii to look at potentially what may be occurring early in infection at the earliest time point by applying some of these epigenetic clocks and looking at these epigenetic changes. I was quite lucky as a postdoc to work on a cohort in Thailand. That was a collaboration between the Thai Red Cross, the US military, and the NIH that essentially enrolled people in Thailand during acute infection and was able to identify people through real-time screening at the earliest possible time point during acute infection. So this spanned from FEBIG 1 all the way up to FEBIG 5. And in this RV254 acute cohort in Thailand, these individuals are placed on antiretroviral therapy at the time point that they're diagnosed and essentially enrolled in the cohort and then followed longitudinally. I believe currently the longest follow-up has been up to 15 years in these participants. And they also are providing optional procedures as enrollment in the cohort, such as leukapheresis, lumbar punctures, brain MRI, and lymph node biopsies as well. In terms of our work, we got quite interested in terms of looking at whether some of these aging signals or epigenetic changes may be occurring at the earliest time point following HIV infection, and whether initiating antiretroviral therapy at the earliest possible time could potentially ameliorate some of these signals or changes that are occurring later in life that we know in these cohorts of people living with HIV on stable antiretroviral therapy for a long time. This was a unique instance where we could go at the very instance where we'd be able to assay potentially what the virus may be doing at that earliest possible time point. And to do this, we essentially utilized flow cytometry and isolated both monocyte cells and CD4 T cells. And then we looked at essentially these biological signals or DNA methylation patterns in these very specific cell types. And in essence, just to eliminate kind of the differences that could arise from cell type heterogeneity. And we also looked at comparison populations of people in Thailand that were not living with HIV, and then also a cohort of people that were on antiretroviral therapy that were living with chronic HIV. In terms of one of the first findings we observed, which is not surprising in terms of the epigenome, was that the majority of these cell type specific changes appeared in monocyte cells compared to the CD4 T cells. Here I'm just showing you the number of changes that occurred that we observed, and essentially over 20,000 of these DNA methylation sites at genes such as interferon and other acute related viral infection related genes were being picked up on these monocyte cells, really supporting, this acute infection that's occurring. To our shock, though, despite kind of these very well-known, drops in CD4 T cell counts, it appeared that molecularly, at least, we did not observe kind of this overwhelming degree of changes in the CD4 T cells. In terms of kind of just some speculation here, it potentially suggests to us, at least at the epigenome level, in the CD4 T cells during this early phase, potentially, these CD4 T cells are kind of the vehicle for viral replication. However, it doesn't appear like they're really an alert sensor in terms of being able to pick up on these very, dramatic changes in terms of methylation changes in CD4 T cells compared to the myeloid cells that we're assessing in this particular design here. In terms of these epigenetic age markers, we had seen that essentially, as you would expect during the acute time point, that the CD4 T cells and the monocytes compared to people in Thailand that were not living with HIV were biologically older, so supporting the initial work and findings from the Horvath group and a lot of other groups showing that this potential epigenetic age acceleration does occur within the CD4 T cell compartment and as well as the monocyte compartment. The other interesting finding, we were able to follow up these participants after 2 years on antiretroviral therapy. Nicely because of the rapid rebound in the majority of the participants within the CD4 T cell compartment and the early initiation of ART. we saw that the biological age versus their chronological age significantly reduced, to around 2 years. This was still older than type people that were living without HIV. However, in the myeloid compartment, we still saw this lingering effect of kind of this age acceleration in the monocyte cells, suggesting potentially that this aging signal, at least in that compartment, may relate to potentially this low-level simmering potential inflammation that may be sitting there in the context of early antiretroviral therapy and viral suppression. But for us, this was kind of the first evidence that this very well-observed phenomenon in people living with HIV seems to occur quite early in terms of kind of this biological aging imprint that we're at least measuring by DNA methylation patterns and these epigenetic clocks in these particular cell types. This work is currently being expanded now across multiple other cell types and into tissues in the cohort to really show potentially where this kind of lingering signal may occur. And we're also looking at participants out past 15 years to kind of track over time potentially what may be occurring as well within this early acute HIV cohort. In terms of building these clocks relevant to retroviruses, this is another thing our group has been working on and has kind of been really focused on looking at deconstructing these epigenetic clocks with the more viral kind of focus. And really, one of the main things we were interested in was that a lot of these clocks, as I had said, had features related to the aging process, but nothing was really tuned to the idea of retroviruses or potentially these endogenous retrovirus activity resurrection that had been observed in the aging field and is now recently, I think, also received a lot of attention. So we asked the question whether we could use information about ancient retroviruses to measure aging. Those that are aware of our genomic material, a large part of it is essentially transposable elements or pieces of DNA that have been inserted into the germline through evolution that have essentially been silenced by DNA methylation patterns. So these lollipop plots here I'm showing you are little circles on the genome are essentially the ones that are dark are areas in the genome that are shut off potentially. And as you can see here in a typical gene in green here, so such as a protein coding gene, at the beginning of the gene or the promoter, there's no methylation patterns, and essentially this allows the gene to be transcribed to make RNA and the protein of interest. And if you put this in the context of the immune system, this also allows a T cell to be a T cell if you look at those T cell related genes that give identity to T cells. But in a large part of the genome, we have a lot of DNA material or sequence that essentially includes elements that are a component of them, human endogenous retroviruses that are conceptually very similar molecularly to HIV as a retrovirus, such that it has two LTRs, it encodes an envelope, and then also essentially can produce particular products if it has not been essentially mutated throughout evolution too much to basically make this a defective and human endogenous retrovirus. But we asked the question of whether we could use these methylation patterns only at these elements to essentially look to see whether this had potentially some link or estimate as a biomarker in the aging process. So to do this work, this was work I completed when I was at Cornell. We looked at a cohort in the US that had essentially received these aging tests or epigenetic DNA methylation assessments. And we focused particularly on areas of the gene that were related to human endogenous retroviruses and lines. We looked at over 12,000 people that had spanned the chronological age from around 18 to over 100 years of age. And then we looked at whether we could train a model using elastic net that could predict, in essence, how old these people were just based on the methylation patterns in their human endogenous retroviruses and line 1 elements in their genome. We did find a signal, and I'm still kind of shocked at how good the signal is. And we reported this and referred to this as retro age. I'm showing you here in the training data set in the 10,000 individuals, their chronological age is on the x-axis here, so around 18 to over 100. And then their predicted age here. So each dot is a person, but fundamentally, if you consider just a linear relationship here, essentially those individuals that are a lot younger could be predicted from those individuals that are a lot older chronologically, just purely based on the retroelement activity proxied through these methylation patterns, suggesting that, these elements are being quite differently regulated or orchestrated through the aging process, and could relate to, I think cell type compositional changes and other potential features. But as I'll show you, we think some of these may actually be involved, particularly in the aging process itself. And then this is just in a training data set looking at 2,000 people randomly out of the initial data set, and then just doing the same prediction based on their DNA methylation patterns. And as you can see, the performance is quite good. So, essentially this concept that there seems to be a very unique biomarker of aging captured by these retro elements that are in people's immune system that we were able to measure. And then we looked to see, well, maybe some of these other papers and groups had shown these features or these particular retro elements in their reported studies, and maybe just not discuss them within the manuscript, or did not know that those were retro elements. This is just an intersection plot showing you essentially that in these retro age clocks that we developed, essentially very few or none overlap with all the existing epigenetic age estimate clocks that are currently being utilized. Really, I think giving support to the idea that, I think these initiatives of using machine learning or prediction models has, I think, high utility. But if you're able to kind of tune these models and have some biological insight or mechanism, and really focus on a question at hand, you can improve essentially, some of the biology that may be occurring using some of the approaches. Some of the other groups just went for kind of what's the best predictor, and then after the fact tried to figure out which of the genes and kind of mechanisms may relate to what they're using to capture. In ours, we kind of did this like informed machine learning approach. We're essentially using what we knew about retroviruses and transposable elements to build kind of a well established epigenetic clock that's proxying the activity of these elements. And then what I'm showing you here is kind of what these have shown us in terms of some of the aging processes. I'll just give one example. So particular elements in the genome here, this is a genomic view of a element that's inserted in the ZK-SCAN3 gene, which is a master transcriptional repressor of autophagy. There's a particular isoform here, so a different version of this transcript that could be expressed and relate to the autophagy process, which is a major mechanism within the aging process. Embedded within here are methylation sites that essentially were captured or used to predict this retroelement age that we captured, and showing that over time in different individuals that are aging, the activity or regulation of these methylation sites is quite different, and this is the signal we're capturing. But we think potentially this relates to something that may be occurring, in terms of mechanisms related to regulating autophagy in this particular example here. And, just showing you kind of the power of using this approach, both from a good biomarker, but also I think to gain some insight in terms of some of these mechanisms with aging that could be targeted or looked at in a future messianistic kind of way. And then lastly, we kind of extended this into the realm of looking at some of these ovulationally conserved data sets that have been published recently. So there was a very nice publication in 2023 in Nature Aging, where they looked at methylation of over 185 mammalian species, and then looked at essentially genomic regions that had conserved cytosine guanines where you could measure methylation patterns. We reutilized this data set and then again trained the model just looking at conserved transposable elements in this data set, and we're able to develop kind of this pan mammalian species clock. Here I'm just showing you those results, showing you that the relative age of all mammals compared to their predicted relative age based on their transposable element activity fit essentially what we were observing in our data set of humans. So really, I think leading support to this idea that transposable elements and potentially retroviruses are essentially quite important in terms of potentially measuring biological aging processes, and really been kind of utilized throughout evolution in the genome as, potential features to silence, to, I think, live symbiotically with, but a subset of them, we think potentially may have actual activity biologically and having impacts on some of these aging-related processes. Um, and then just to summarize this line of work, as you could imagine, there's a large literature in terms of looking at transposable elements, human endogenous retroviruses in the aging field. And it's really been, I think, reemerging as kind of a key focus for the geroscience community. Two papers here I'm showing you, just kind of leading support to kind of this idea, this resurrection of endogenous retroviruses during aging, potentially reinforcing senescence. In one manuscript in Cell recently. And then it was nice to see after our publication, a subsequent publication from a well-known group that has been looking at transposable elements in different species and mammals, showing in another US cohort that transposable element methylation states in blood cells predict age and disease. So, it was quite nice to see that essentially they were able to confirm some of our epigenetic work in a different cohort, just leading kind of support to this idea that these transposable elements are potentially being altered or changed throughout the aging process. And then just to end, so these really have been kind of recently considered from the aging field, at least from experts looking at biomarkers to look at in the terms of intervention studies, in terms of the recommendations that should be used in human intervention studies. Here I'm showing you well-known supported things, at least, from the aging and geriatric side of the equation, such as gait speed and hand grip strength. Looking at cognition, such as with the, Montreal Cognitive Assessment Test. But here you can see that one of the major included biomarkers has been in the inclusion of the DNA methylation epigenetic clocks to these human intervention studies. Really, I think a large part of this is because of the amount of information that could be gained from, a simple test looking at DNA that really, I think allows you to span quite a lot of different algorithms and estimates and kind of get ideas in terms of what may be modified in some of these human intervention trials that are ongoing. But to move to the last part of the talk today, I really wanted to, I think, emphasize one of the areas we're spending a lot of time in terms of collaborating and learning from those that have led a lot of the, I think, really rigorous and quite arduous HIV clinical trials. And really the idea being here is to leverage these clinical trials in people living with HIV for post hoc analyses to see whether trials that essentially, quote unquote, I think failed for their main outcome of measure actually could be repurposed by accessing biospecimens and looking at aging biomarkers. And some of these indications to inform the aging field, which is quite interested in a myriad of different approaches and therapeutics. I would say the overwhelming majority of it in the current environment we're seeing in terms of this very chaotic, I think, attack on science. And, I think the evidence in the aging field suggests a lot of it is hype, and kind of not well supported from the evidence in terms of rigorous clinical trial design. But our attempt really has been to go back and to get at least some evidence to say, yes, in terms of pharmacological approaches, there seems to be some impact in any aging biomarker to suggest that, hey, maybe a better designed aging focused trial needs to be pursued, or no, we don't see activity there. And, potentially caution is warranted to kind of suggest that maybe these interventions are not moving the needle sufficiently to give evidence to kind of design these more rigorous trials to pursue. But here I'm just giving examples, and those that are on the call are well aware of, all the different approaches essentially that have been tried in terms of clinical trials in people living with HIV. But, in terms of the biospecimen availability, I think this is an incredible opportunity to leverage samples that are essentially sitting in the freezers to gain insight into some of the mechanisms that could inform both the geroscience field, but more importantly, people living with HIV in terms of what potential things may be having an impact on their aging process. One of the ones we've started with, I just included this because this was data we're recently wrapping up and really building on, is looking at GLP-1 agonists as a therapeutic. It's kind of emerged as kind of the most, I think, talked about therapeutic for both the aging process. Cardiometabolic disease was the main indication, and it seems like, GLP-1 seems to be in the lay press, but also, I think among different areas of science, kind of the treatment for everything. We got quite intrigued in it, or I had been following this field for a while from my time in Hawaii, mainly because I had seen some initial data suggesting, GLP-1s in people in Hawaii that had high BMIs, in terms of being able to have a large impact in terms of losing weight there. But I've been more interested in terms of looking at it from the aging side of the equation to see whether it had any potential impact in terms of some of these aging markers that we had been measuring. So to do this, we've kind of been working with colleagues in the HIV field that had run GLP-1 studies, both from the ACTG and then also NIH funded. Here I'll show you a collaboration we have with Grace McCumsey's group, who had a early study that was funded to look at a randomized double-blind placebo-controlled trial of semaglutide and then a placebo. And looking at the impact on total and visceral adipose tissue quantity in regards to HIV-associated lipohypertrophy. And then we performed a secondary analysis where we looked at the effect of semaglutide on epigenetic aging biomarkers in participants both in the placebo arm and the treatment arm over 32 weeks. So at that week 0 time point and then the week 32 time point. And the general hypothesis was essentially that there could be potential pleiotropic benefits of semaglutide on biological aging. So really just to say, okay, we're using these aging markers that the aging field is using for all these dietary interventions, the ones that are being performed for looking at exercise, drug therapeutics. We have here a very rigorous design where we're looking at a placebo group over time and then a group essentially that is receiving semaglutide, and just to see if there were any signal that may be occurring. These are the baseline participant characteristics broken down by all participants, all 84, and then after randomization into the semaglutide arm and the placebo arm. All of the criteria essentially were in line with the main outcome of measure for the trial. So as you can imagine here, elevated BMI. There was a slight difference in terms of the C-reactive protein in the plasma biomarkers that were assessed, a little bit elevated in the placebo group on the median compared to the semaglutide arm. But overall, all participants were pretty well matched across, after randomization. We looked at their initial baseline aging rates using some of these clocks and then also our retroelement age. As expected, according to the clocks that measure chronological age, mortality risk, and then the retroelement clock, we saw an increase compared to their actual chronological age, or an elevation in mortality risk by up to 9 years in both participants in the placebo and the semaglutide arm before randomization, and then a greater than 10-year acceleration in terms of the retroelement age, suggesting that these transposable element methylation patterns are potentially quite different in these individuals based on their chronological age compared to participants in the general population. This is just summarizing very briefly what we've reported, and this is currently under review, looking at this broad deceleration in epigenetic clocks across multiple epigenetic clocks in the semaglutide arm compared to the placebo arm, suggesting potentially, some impact here on aging. We had corrected in these models for essentially adiposity changes and inflammatory changes. So it seems like some of these changes are still maintained despite the changes that may be occurring due to adiposity and inflammation. Across the majority of these clocks, it appears that there's a 3-year difference over the annual rate over the 32-week period in the semaglutide arm compared to the placebo arm. So just comparing the difference in participants over that period of time, it looks like those participants in the placebo arm are aging quicker based on this biological age estimate than those individuals that had received semaglutide. So, we're essentially not making people younger here, but just reducing kind of this age acceleration that may be occurring over the period of time that people are assessed. And then when you looked at the actual pace of aging, so this was kind of that speed accelerometer estimate I had told you about previously, it looked like it was about 9% slower in the semaglutide arm compared to the placebo. Interestingly, a recent development using this intrinsic capacity measure developed by the World Health Organization and included in a epigenetic clock developed by the Buck Institute up north. We didn't see any real signal there in that particular estimate. But we did, across multiple clocks kind of see this aging signal, kind of telling us, it seems like some of these effects we're seeing in terms of all these reports about GLP-1 benefiting all these different disease indications, this kind of fits that hypothesis that there seems to be some impact here, at least on some of these aging markers, suggesting that, it may have these kind of broad effects. Um, and then we've extended this using these estimates that can span different organs. So here I'm just showing you the application of this systems clock that broke down estimates of organ age according to clinical measures of factors that relate to different things such as the kidney age, the heart age, liver age, brain age by utilizing protein markers in their estimates. And as you can see here, again, semaglutide conferred multi-organ impacts across these different age-related estimates, reducing essentially the estimates compared to the placebo group over the period of the trial. So I think just gaining further support here, it doesn't seem to be purely a phenomenon that's impacting just the immune system or inflammation per se, but seeming to have broad effects across the body. And I think really gaining kind of the first support here that, there is some evidence that, I think these more rigorous studies in the GLP-1 space that a lot of groups throughout the US are actively pursuing currently, I think do have evidence to really proceed. And, hopefully we find that those studies also confirm some of these results we've observed in people living with HIV. And then we've also looked at another study in the ACTG, A5371, the slim liver study that Jordan Lake and Christine Erlinson and others in the A5371 team had led. And again, repurposing a study that was initially designed to look at MASLD in people living with HIV and utilizing PBMC samples at week 0 and week 24. And then looking essentially at whether the aging markers over time may relate to those individuals potentially that had the greatest change in the primary outcome of interest, which was the quantification essentially of liver fat over the period of time, which, in this trial, it was very successful in the sense that, this really, I think, supported GLP-1s being utilized, to treat liver disease, both within people living with HIV and the general population. But for us, another opportunity here to quite rapidly repurpose and then gain additional evidence suggesting that, hey, maybe some of these aging-related processes and markers may help kind of inform the application of GLP-1 for disease indications. And then this is just summarizing, what the trial had observed in terms of a greater than 30% reduction in IHTG in over 58% of the participants. And then 29% of the participants in this report had complete resolution. And then they did observe a suggestion that potentially some of these improvements more greater in women living with HIV and participants that were older seem to have a greater benefit in this initial look of this report. Just to summarize what we've seen, and again, this is another publication currently under review. Participants with beneficial changes in epigenetic age biomarkers showed a significantly greater reduction in IHTG and physical function measures. So here, just showing you those individuals essentially that were showing improvements in their aging process were those individuals essentially clinically that were showing the greatest percent change or decrease in IHTG. And then also looking at markers related to telomere length that were proxied from these epigenetic age markers. These were individuals that also showed some change in terms of some physical function, so essentially having a greater chair rise speed in the particular context of this single-arm trial. So, in this particular trial, one of the limitations, we did not have the placebo arm to compare to, but at least in terms of comparing those individuals that were kind of responders versus non-responders, and we had some evidence here that, these markers were kind of informative to be able to potentially understand that. And then lastly today, this is something we're currently working on with Steve Greenspoon and the Reprieve team. We've looked at the epigenetic clock biomarkers in the context of Reprieve. So for those on the call that are not aware, Reprieve essentially was a randomized trial conducted in people living with HIV and antiretroviral therapy with low to moderate ASCVD risk, given essentially either placebo or pivastatin, and then looking at the outcome measure of first primary MACE and the remarkable success of Reprieve, showing essentially that the trial had to be stopped early because of efficacy and has really changed clinical practice in terms of the prescription of pivastatin to people living with HIV or statins as a mechanism potentially to prevent cardiovascular disease outcomes, despite not having typical indications to receive a statin. But we got quite interested to see whether potentially there was any aging-related changes that could be studied in the context of reprieve. So some of the questions we're currently working on, do younger people at risk for CVD that are living with HIV demonstrate advanced biological aging? Do statins alter advanced biological aging processes either by reducing or preventing ongoing changes? And one of the other questions we're currently asking within Reprieve is, will the effects on biological age help explain reductions in cardiovascular disease events and vulnerable plaques seen in Reprieve? We're quite excited to pursue this study. We did have a pilot look, a very small study where we looked at individuals, around 100 individuals in the placebo and the treatment arm. And we did see increased biological age among these low to moderate risk CVD risk people living with HIV that were enrolled in Reprieve in the US. Here I'm just showing you the difference between their chronological age and this mortality grim age marker. As you can see at entry, everyone above the line. So despite having low to moderate CVD risk, individuals in Reprieve were biologically older by this estimate. And then we've observed in a small follow-up study where we looked over 24 months, some initial pilot data giving us support to do the whole subset of Reprieve for profiling that pivastatin potentially could be having an impact on the aging process or these aging biomarkers in these individuals over the 24-month period within Reprieve. Here I'm just putting the difference that we've observed in Reprieve for this pace of aging measure on the pivastatin arm compared to placebo in blue here in benchmarking to a well-known aging paper that used the same exact measure but had looked at healthy people that were undergoing a caloric restriction or an ad lib diet. And just benchmarking essentially the difference between the statin and placebo arm compared to what was observed versus caloric restriction or ad lib eating in this calorie trial that had been looked at from the Geroscience field. The main difference here in the calorie trial, people were age 38. They were so they were not living with HIV. They were non-obese. This is kind of the remarkable difference that was reported in terms of some of these changes now that are being followed up for looking at dietary restriction as a potential aging therapeutic approach. And then within Reprieve, at least in this very small pilot study, we've observed, and these are people median age 50, living with HIV, and have low to moderate CVD risk. And we're quite excited to extend this to, a more rigorous sample size to really, see potentially if this initial finding holds up within Reprieve.
Hey, Michael, just wanted to tell you 849.
Okay, thanks. This is the last slide, I believe. And then currently, we're also collaborating with the ACTG to harness clinical trials, as I had said, that were not initially designed to target the aging process, but to repurpose these trials and samples in the repository to look at some of these differences over time. One of the most studied within basic mechanisms and looking at animal models and the aging process in general has been rapamycin. We hope to do a post-hoc analysis of the rapamycin A5337 trial to look whether there's any impact there on some of these aging processes. Growth hormone, ruxolitinib, methotrexate, and then also looking at the maraviroc trial as well. Really just to kind of go across the gamut of different trials and to see whether we see indications. And just to end, in terms of a lot of our other work, which I was not able to present today, one of the remarkable things we have observed is it's quite stable in terms of these aging markers. So we have never seen, at least to this point, evidence across all the myriad of HIV clinical trial data we've looked at, such as HDAC inhibitors and other potential therapeutics that are making people age quicker. So the good news is we're not essentially making people age faster by doing some of these therapeutics. The hard thing has been a lot of the data we've looked at has been negative data. So nothing's really moving the aging process. But the most popular suspects that people have been talking about, such as GLP-1, as I presented, and other things that relate to metabolism, I think do move the needle quite dramatically. So those are the things that, we really feel quite confident that by having a span across all the therapeutics, 90% of them aren't going to move the needle. Things are quite stable, which is good news. We're pretty resilient to a lot of these interventions. Whether these were why these interventions failed, this is unknown. But I feel quite strongly that at least if the needle is moving in a positive direction, that's quite good evidence that that's something that really needs to be followed up on and studied more intently. So just to summarize today, hopefully today I built out some background in terms of the geroscience field and our research looking at people living with HIV, exhibiting an earlier onset of age-related diseases and increased biological aging, really positioning HIV and, more importantly, those in the community to really advocate for research that is focused on aging mechanisms and the aging process. And then secondly, we're really interested in collaborating with groups that are interested in terms of looking at the interplay or relationship between HIV biology as an exogenous retrovirus and endogenous ancient viral elements such as human endogenous retroviruses and line ones in our DNA linked to aging mechanisms. I didn't have time today, but there's a lot of beautiful work that was conducted very early on, basic mechanisms showing that the HIV machinery activates the human endogenous retrovirus activity. Also in regards to T cell responses, having particular T cells which are responding to human endogenous retrovirus. That biology, a lot of discussion should occur and needs to occur between those that have been doing that from the HIV field, but also those that are interested in looking at these endogenous retroviruses as well. And then lastly, I really think the biggest opportunity for us in the field is really leveraging these completed clinical trials in people with HIV with a geroscience guided approach as a post hoc analysis to inform the repurposing of FDA approved therapeutics for improving health span. And as potential future studies are designed or planned to target the aging process in people with HIV, I think having this data would be quite helpful in terms of saying, these are the measures we need to include in a follow-up study with a more aging focused outcome. But also, I think give kind of some insight into where resources should be dedicated, given that, I think now resources are becoming quite limited to be able to conduct clinical trials. But to end, I'd just like to acknowledge, all the groups I've had the opportunity to work with, the NIH, and then most importantly, I think here being at UCSD, I'm quite excited, I think to meet others that I've not met yet, and I think plug into the incredible infrastructure and history here that has occurred both within the clinical trial space, basic mechanism, but more importantly, just being a wonderful place to be back home. So thank you so much.
I've started to do loud claps so you can hear it, not just virtual ones, but um I have a feeling there are going to be questions from the group, and I'll give people time to gather their thoughts. There was a question though that came through while you were talking about the GLP-1s. And Katie, I don't know if you just want to ask your question. Thank you. You are showing your camera. Go ahead, Katie. I think you're muted, but it doesn't look like you're muted, but we can't hear you. Okay.
It was in the chat or?
It is in the chat, but she may have more, and yeah. So she was asking how studies differentiate the different mechanisms of benefit from GLP-1s. So, how do you know what the mechanisms that are leading to the benefit as far as improvements in aging?
Yeah, so currently, I think a large part of the effort has kind of been in this microdosing space, which has been, I think, pursued anecdotally by people. But I do know of a few studies that are being designed in terms of low dose GLP-1s in people that typically would not have your classical indications for receiving GLP-1 for either weight loss or other features. And then the other thing that currently is ongoing too, in terms of looking at which cell types are expressing GLP-1, we have a little bit of data, and it's in the literature as well, suggesting that Treg cells, which recently were awarded the Nobel Prize, express the GLP-1 receptors. So it does seem potentially some of these may be impacting target tissue organs that express GLP-1, but also potentially the immune system itself might kind of have some regulatory activity that might be modified by GLP-1. But the exciting thing I think there is, it's kind of a field that everyone's working in that space now. So we'll see a lot of great data very soon. We really went at it mainly just to see, was there any effect in terms of completed work that had ongoing from, rigorous clinical trials to see whether there was evidence there. And that was kind of our first suggestion that, hey, there was signal there that warrants, all these groups kind of moving in that direction and getting funding and data to really, I think, build out more insights into that field. So, 'cause a lot of people are on those drugs. So, the more we know, the better.
Katie, I don't know if you wanted to ask or add anything else.
Oh, yeah. Can you hear me now?
Yeah.
Mhm.
Laptop microphone doesn't work anymore. So, hi. I'll do great.
Katie, you're cutting in and out, unfortunately. I know.
For
You
mine.
may want to just
I'll
turn your
go with
camera
thank
off.
you.
It's unfortunate, but
I'll go with thank you. Any better? Fine. Thanks, Michael.
Sorry.
was
Yeah.
that maybe that's it? Okay. I know there are lots of other folks who are in this space. Dan.
Yeah, great talk, Michael. I think Jill, you may not have asked Paloma's question. There
Oh,
was a
I'm
question
sorry.
about planning. Are you planning to compare it to other GLP-1s like tirzepatide?
Yeah, we're hoping to do that. I think the hard part I think the comparison we have currently, we've mainly studied GLP-1 in the context of people living with HIV. The tirzepatide collaboration I have currently is going to be in people without HIV. So, whether that comparison, is like fair comparison, I think I'd be a little bit cautious on that comparison. I do believe some of the other groups that are working in this space potentially have an opportunity to do that. One of the major shifts, at least I've observed, just kind of, tangentially following kind of discussions and seeing groups present different data. Definitely Novo Nordisk has become more interested in terms of aging. They had zero interest, I think, early on in terms of looking at this. It was understood, I think, because they were really trying to just really market it towards a particular indication. And, they had zero interest beyond that. Now I think they've moved in that direction. And I've heard, at least through the grapevine, Eli Lilly is now kind of interested from research side to also pursue that. But, again, I think they've been slowly kind of doing that, but definitely that would be something both of those players would be quite interested in a head-to-head comparison. I do know tirzepatide, right, is kind of the one that is receiving a lot more favorable attention in terms of, efficacy, side effects, low dosing. So it's definitely the preferred in terms of if you give people an option from that kind of landscape. But from a rigorous comparison in the aging context, I'm not currently having the structure to do that, but it is something that needs to occur.
Yeah, I think it's been interesting. I know, with the tirzepatide, certainly it's been more efficacious in terms of the weight loss, but I did see that there was some data in regards to cardiovascular risk that, at least it looked like semaglutide was the only one that did show some, I think, cardiovascular benefit versus tirzepatide did not, which was kind of a little bit odd as to why there's I guess some difference between the two different ones, although I think tirzepatide is a dual GLP GIP. So but anyway, thank you for your work. If you're interested, so myself and Paloma we're on the clinical side at Owen Clinic in terms of doing the weight management stuff. So we actually are putting people on GLPs. We don't have anything going on prospectively, but if that is of interest, we should probably talk, because we're probably the ones to
No,
work
I'd
with
be
you
happy to
on that.
follow up. And then, I today don't have the finished data, but we also have a very nice collaboration with Miley Karis as well, looking at a cohort at UCSD, as well. So that's something, we'd be quite interested in terms of looking at, because one of the other interesting features, in terms of probably what you've seen clinically as well, in terms of kind of this responder, non-responder question as
Yeah.
well. And then, essentially from the weight management side, keep upping the dose until you get your effect. And then, from the aging side, the question is how low can you go and how much do you need to take in order to get any kind of aging benefit? Currently, that's kind of the discussion ongoing. And I think that's an interesting point too, in terms of if you look at that from the aging field and what they've been doing with the sirolimus in terms of low dosing, intermittent, kind of taking that with no real evidence. I think the GLP-1 space similarly is going to emerge in that context. Like, do you take it once a week? Do you take, lowest dose? But yeah, I think, having kind of the insight from the clinical side, definitely is something that needs to be incorporated, because clinically, it's like, yeah, it's, you do your best. But no, thank you so much.
We'll be in touch then.
Yeah.
Thanks.
I was muted. Thank you so much. It's past 9 o'clock and we try to respect everyone's time. I had some questions about the NRTIs and telomeres, but I can talk to you about that offline. Just something that's bringing me back to fellowship, which was like the hot topic then that doesn't seem to be phasing anyone anymore.
Yeah, I didn't have time to go into that. I mean, that's another
Yeah.
space we're very interested in terms of,
Yeah.
the aging field is 3TC actually is a major aging therapeutic being pursued. So, that's
Oh,
another.
interesting. Okay.
Yeah,
Well, we'll have to have you back for more.
Yeah, but
Thank you so much,
no, thank you.
Michael.
Everyone have a good Friday and good weekend. So thank you for the
Happy
invitation.
Friday, everybody.
Bye.
Bye-bye.